Hello class, and welcome back for our sixth lecture video here for Unit 6. Today's topic is going to be on the process of cell division, in particular mitosis as well as cytokinesis. So what we're looking at right now on this screen would actually in fact be plant cells, not animal cells. One of the dead giveaways is this rectangular structure that we can see here, as well as these thick clear barriers which would actually represent the cell wall. Right, when we look at cells underneath a microscope, one of the easiest ways to figure out on whether or not they're actually in some particular stage of cell division is, do we see their chromosomes that become visible? And we could see that in this slide right here, as well as this slide or cell, I should say, right here. All right, because we can see these thick bands of chromosomes. Otherwise, every other cell that we see, we're seeing just a large overall nucleus, which is a cell that's currently in interphase. Now, if you remember, cells should spend at least around 2/3 of their life in interphase, and we should see their DNA as chromatin, as we do in all these other cells here. So let's proceed through and start to learn about the different stages of mitosis. So first off, the definition of mitosis would simply be the division of a cell's nucleus. That's the key. Because remember, by this point, all DNA has already been duplicated during the S phase of interphase. So at this point, we're just trying to take the contents of the nucleus, divide it in half into two brand new nuclei. Later on, we'll go through full-fledged and finish out cell division to get two identical daughter cells. So just basic definition of mitosis is the division of a cell's nucleus. All eukaryotic cells will perform mitosis. Do not confuse that, though, with binary fission that we discussed that bacteria do, which is a form of asexual reproduction. That is straight up cloning, where the DNA is just copied and the cell just instantly kind of vibrates and splits in two. Now, mitosis technically is cloning also, but all the steps that go into it, copying organelles, getting the DNA copied, splitting it, it's a lot more detail-oriented than binary fission is, so it's a different class of cell division altogether. So we talked about a lot of organelles earlier in the year with our cell organelle unit, but now we're going to come across one that I didn't discuss and we saved for this point. And they're called centrioles, or sometimes referred to as centrosomes. And here's what they look like. Every cell starts off with at least a pair of them, and it looks like two little ziti noodles that you would eat for dinner. All right, if you're Italian, you got to know what ziti is, right? Come on. So, but if you look at this picture, it looks like they're little straws that are just kind of all glued together into a big cylinder. All right, what these centrioles are made of are actually microtubules, which were one of the three cytoskeleton parts we discussed earlier in the year. And microtubules are actually the largest of the three. The other two were intermediate filaments and microfilaments. So these two centrioles, which are made up of microtubules, they're going to be seen shooting out what are referred to as the spindle fibers that you should have seen in 7th grade with the process of mitosis, and we'll get to that and you'll see them again in a moment. Now, there are two key differences in how mitosis goes down in both animal and plant cells. The first of the two are the fact that we only find centrioles here in animal cells. Plant cells will not have them because they're going to make use of their cell wall and cellulose fibers that they actually have that we don't, that are then used to move chromosomes around and eventually split the cell in two. But you and I don't have a cell wall or cellulose, so we're going to use centrioles. All right, so as we're going to see, for complete cell division, we're going to go through both mitosis and cytokinesis. So it generally starts with a cell here where you see a big solid nucleus. Eventually the chromosomes start to condense and you start to see them underneath the microscope. The chromosomes line up along the middle of the cell, eventually split, and here you can see the formation of two brand new nuclei. See how it's starting to get really dark blue like we started off initially? That would actually be the end of mitosis in telophase or telophase, however you want to pronounce it. And the last thing that'll need to happen is we need to split the cell into two brand new ones, which would be cytokinesis. So let's start with the first stage of mitosis, which is prophase. Now, what I want to remind you is all the DNA, all the chromosomes that a cell has, have already been duplicated before we even get to prophase. So for you and I, all 46 of our chromosomes would have already been duplicated into 46 sister chromatids. Remember, count your centromere, so I would still just have 46 chromosomes as long as they're in the X shape. But we don't even ever see these chromosomes until we hit prophase, where all the DNA, all the loose chromatin, remember it looks like a big bowl of spaghetti or lo mein noodles, coil up, coil up, coil up until you get these X shapes here that you see. Excuse me. And then the other two key things that'll happen during prophase are the centrioles, which have already been duplicated during G2 of interphase, will move the opposite ends of the cell. And the nucleus, so you can see that happening over here, the centrioles move to the poles, and the nuclear membrane or envelope broke down. The chromosomes here are starting to condense into that X shape that you can see. All right, so we let the animation here continue momentarily. Again, every chromosome you now see as a sister chromatid pair, which is comprised of two chromatids connected by a single centromere. Count your centromeres. There goes your centrioles moving to the poles, the nucleus breaks down, and again your chromatin condenses into however many chromosomes that cell normally has of its diploid number. All right, next up is going to be metaphase. Before I get there, I just wanted to show you again a picture. This would be a cell in interphase. You can actually see the DNA as chromatin, so it looks like one big solid nucleus. But as soon as the nuclear envelope breaks down and the chromosomes start to condense, you can start to see chromosomes starting to form. Now next up is metaphase. Meta stands for middle. So what's going to happen now is the microtubules or spindle fibers that are being shot out by the centrioles. So if you look down in the animation, here's your centrioles I'm pointing to. All these little lines here would be spindle fibers. These yellow lines here in this cell would be spindle fibers, or these green lines here would be spindle fibers. They're going to attach not to the centromere, but a very specific part of the centromere called the kinetochore. So that'd be these little two orange spaces that you see on each side of this sister chromatid pair. Those aren't two centromeres. There's only ever one centromere for every X. But if you see two little dots, those are the kinetochores on each side of it. So if the centromere is a deadbolt on your front door at home, the keyhole where the key actually goes into and latches into would be the kinetochores. That's where the spindle fibers attach. And then, once they're attached, the centrioles are going to push or pull all of the chromosomes or sister chromatid pairs to line up right down the middle of the cell in single file line. It doesn't matter which chromosome is in which space. It's not like you're lining up tallest to shortest or shortest to tallest. It's just randomly get in line like you did in elementary school to go to the bathroom, right? Everybody lines up before the teacher brings you to the restroom. But they all have to be lined up one after the other like you see here, and that's done by the spindle fibers attaching to the kinetochore. And again, you see that in the animation as well over here. Next up is the one that a lot of students every year get a little bit confused with, and it's anaphase. At this point, what happens is all the spindle fibers that are attached to those kinetochores, all right, they start getting pulled back by the centriole. So down here in the animation, the centrioles, it's like a tug of war. They start pulling on their spindle fibers. And as that happens, it rips apart the sister chromatid pairs into two chromatids. So in anaphase, sister chromatid pairs no longer exist because they're getting ripped apart. All right, so sister chromatid pairs will split with one chromatid, or you could say one chromosome, moving in opposite directions of the cell. And since the sister chromatid pairs are splitting, your chromosome number is going to temporarily double due to the fact now that every chromosome or chromatid has its own centromere that I'm pointing to, like the little yellow parts at the end. All right, so if I originally had 4 X's in metaphase, I would have had 4 chromosomes. But now I have 1, 2, 3, 4, 5, 6, 7, 8 chromosomes. Sorry about the bell behind us. So again, 8 chromosomes or 8 chromatids is what I would have total here. The answer would be 0 sister chromatid pairs since they've been split. And now we want to finish up mitosis with the last phase, which is telophase or telophase. At this point, you're going to start to see two new nuclei, which is the plural for nucleuses, all right, begin to form. And you can see that over here in the animation. There's your 4 chromosomes in each cell. See how the nuclei now start to reform to surround them? And you can also see that those 4 chromosomes are now going to start to uncoil back in the chromatin, and you start to see a big solid blue purplish area inside, correct? That marks the end of mitosis. It's officially complete at this point because you have 2 brand new nuclei that you split from the one original. Now, as far as chromosome number goes, remember in Anaphase, all right, if I go back real fast, I told you that your chromosome number temporarily doubles here. Well, it's going to go back to the original diploid number now in this instance, which is going to be 4. Once the new nuclei form, just give me the chromosome number that you would see in one specific nuclei, not both. So now if I said how many chromosomes would you see in this cell in telophase, give me the number 4 again, not 8, because technically these 2 nuclei are their own cells. We just got to go through that last process of cytokinesis to get them into being 2 separate daughter cells. So telophase is complete once I get my 2 new nuclei. What you'll also start to see in some cells is something called a cleavage furrow, where the cell starts to pinch in on each side, and you start to see this like kind of little fission lines start to happen between the 2. That's the preparation for the last stage of division, not mitosis, but division, which is cytokinesis, once you start to see this cleavage furrow begin to form. And again, here's an electron microscope of what it would really look like in real life for a cell. Literally looks like it's literally getting pulled in. It's like 2 water balloons and something's pulling in the one edge of it to try to separate it into 2. Well, to do that, we have to go through the final stage of division again that I call, or that scientists call, cytokinesis, which is officially the full separation of the 2 daughter cells by cutting the cytoplasm in half. And you look down here, here's your cleavage furrow, pulling, and eventually, boom, there's your 2 brand new separate daughter cells. So your second key difference in mitosis between animals and plants is going to be only animals do this cleavage furrow and separate the 2 daughter cells into 2 individual units. So this is only done by animal cells, because this is being aided, this pinching is being aided by the centrioles and the spindle fibers that are within the cells. Plant cells do not have centrioles, as I mentioned earlier. So what they do to separate the 2 cells in half is really they just build a new wall right down the middle of them, sometimes also called a cell plate, to separate the 2 daughter cells into 2 complete different entities. But they never fully separate into 2 separate units. You're going to see on the next slide that they're still connected down the middle by a cell wall. And the reason for that is they don't have a skeleton like you and I do. They need every last bit of strength that they can, so they want to stay connected at least in some fashion together, and that's going to be by the cell wall. So if you look here, here's what's happening right now. So here would be anaphase, all right, of a cell, a plant cell, and here's the creation of that cell wall or cell plate you can see down the middle. And then once it is fully built, you have 2 daughter cells here where my arrow is. But you see how they're connected together still by a thick cell wall for strength? So think of each cell, top and bottom, as 2 bricks that are still connected by a cement line, especially if you have a brick wall at home on your home. While animal cells, cleavage furrow forms, pinch away the cell membrane into 2 separate, completely separated animal cells that you can see here. And at that point, we are done mitosis. Remember, the cell cycle is made up of both interphase and all the stages of mitosis, PMAT, as well as cytokinesis, to lead you to 2 identical daughter cells that have the same number of chromosomes as the original parent cell did.